All right. Thank you, everyone, for joining this session of the Bioconductor Africa Seminar Series. We're so happy to have you join us, and especially if you're joining from East Africa. I know it's quite late in the evening, but thank you for sticking around and coming to this session. In case you've not seen me before, hi, my name is Nyasita Ondari, and I organize and lead the Bioconductor Africa Seminar Initiative to spread bioconductor use on the continent of Africa. So I'll just read a few housekeeping rules and then hand it over to Stephanie, who's going to be our presenter today. And our topic today is going to be GWAS data analysis using the Genesis package. So I reckon if you're on this call, you probably work with SNP data or GWAS data of some kind. So we are happy to have you here. So if you look at the Frama pad that I just posted on the chat, we have a code of conduct at Bioconductor. You can scroll to line 16 to read it in full, but the TLDR is if you experience or witness unacceptable behavior, which may include harassment, intimidation, bullying, or any kind of unacceptable behavior, please feel free to message me during this call or also write to Maria, who will join us a bit later. So we will be recording this call and I will be uploading it to the Bioconductor YouTube channel. So if you'd not like to be, identified either by your name or organization, please feel free to anonymize yourself. You can use a pseudonym or an emoji if you don't want to be included. Yeah. We will also be removing AI note takers from this session, unfortunately, for privacy and safety reasons, and also because we will make the notes and recording available so you can be able to follow this at a later time. So in case you have not taken part in the roll call again, please feel free to scroll now to line 31, and you can add your name after line 40. Yeah. So I will introduce Stephanie and then hand it over for her to lead this session. So Stephanie has a background in astrophysics and over 15 years of experience in statistical genomics and genetics. She has extensive expertise in analyzing large-scale genomic datasets and developing scientific software, and is the author of several R and bioconductor packages and Python-based pipelines for phenotype association testing. If you want to read the full bio, you can scroll to line 44 of the Frama pad and look through. So I will stop sharing this. Hopefully you've been able to scan to join the mailing list or the Zulip channel. And yeah, thank you so much everyone for joining. And I will hand it over to you, Stephanie. Thank you.
Everyone, hello. I'm Stephanie Gogarten. I'm from the University of Washington in Seattle, where it's 8 o'clock in the morning. So thank you all for staying up a little bit late in your time zones, so we could make this workshop happen. I'm going to share my screen, and this is the course website. So this has all the material that I'm going to be covering today and next week on it. So you can feel free to look at this on your own time and spend a little more time going through the R code that I'm going to cover. Uh, and there's also some exercises that for each of the sections that I'm not going to go through, but they're for you to try on your own later to get a better idea of and practice of how to use the software. So I'm going to start with an intro to the genomic data structure format for genotype data. And we're starting here because this is the data format that Genesis relies on to store genetic data for analysis. And so in order to use Genesis, we need to understand how the data is stored. Excuse me. So we're going to learn about genetic data storage. We're also going to learn how to convert variant call format files to GDS and explore the data in R. So if you've worked with SNP or whole genome sequencing data before, you've probably seen at least some of these files formats. Uh, One of the most common file formats now is the variant call format, and it also has a binary form. You may also have worked with Plink files before, so there's several different types of Plink files from the very first iteration of Plink files, which were PED files, through bed files, and now Plink 2 has pgen files. Um, and so all of these data storage formats encode the same information, they just encode it in different ways. And we're going to see how to convert from these formats into the genomic data structure format, which is the format we're going to be working with in this workshop. Uh, GDS is built on a C++ library called Core Array, and it's a generic data container with a hierarchical structure. It can efficiently store multiple array-oriented data sets in a single file. Um, it was originally designed for SNP array data, and there's another R package called seq array, which was built on top of the original GDS format to extend to sequencing data. And we're going to be working with this R package, the seq array R package, which with the structure of GDS that it defines. So why are we using this file format? It contains all the same information as a VCF file, and we can convert the VCF file directly into GDS. But then this file format is integrated with R, um, and there are packages that provide the same analysis capabilities as using VCF, but you can easily write custom analyses to analyze your data using the storage format. And the file sizes are generally much smaller than VCF, and it's faster to read data out of VCF. And it's also very efficient to access subsets of the data without reading the entire file into memory. Um, this is a collection of R packages that are all built around the genomic data structure. And it allows us to efficiently retrieve data and use it in R. So there's GDS FMT, which is written by Zhuwen Zhang, who's the author of the GDS format and related packages, which is the core library that allows us to connect to GDS files. There's seq array, which is the data format that allows us to connect to sequence data, but you can also use seq array for storing SNP data from arrays as well. Um, there's seq var tools, which is a package that I wrote that adds additional functions on top of seq array for some common tasks like displaying genotypes and calculating summary statistics. There's a package called sniprelate, which is a package that allows you to do relatedness and principal component analysis. And then finally, there's Genesis, which is in the title of this workshop, which allows you to do association testing with mixed models. And Genesis relies on all of these other packages in order to do what it does. So this is a brief schematic of the seq array GDS format. So we have the GDS file, which is stored on disk, and then the C++ library that allows us to interface with it. That's called by the GDS format R package. Seq array provides an interface between all of these other R packages like sniprelate and Genesis and the GDS format data. Um, so to explain a little bit about how data is stored in a seq array GDS file, this is what a VCF text file looks like. You might have seen one of these if you've been working with sequencing variant data. And it stores each information about each variant in a single row. Uh, and what that means is that every time you want to retrieve information about a specific type of data, like if you want to read the chromosome for every single variant in your file, or if you want to read the genotypes for every single variant in your file, you have to scan through the entire file in order to extract that information from every row in your file. And so what seq array and GDS does is it takes each of these different types of data and it stores them as arrays contiguous to each other in the file. So it'll take all of the chromosome data and store it next to each other in the chromosome field of the GDS file. And the same for the position and the ID, etc. It stores the reference and alternate alleles in a single allele array where it lists has comma separated lists of all of the potential alleles, which could include multi-allelic variants. Uh, it decomposes the info field. So if you have multiple annotations in your info field, each of these will be in its own separate array. And then finally, it'll take the genotypes from your file and store it in a three-dimensional array, where the first dimension of the array is your allele, and then the next two dimensions are sample and variant. And this makes it a lot faster to pull out just a specific type of information for the file. So if you just want to read the genotypes, you don't have to scan through the entire file. You only have to go to the array that contains the genotypes and pull out that information. And this is just a view of what it looks like if you print out information about a GDS file in your R session. And these are all the nodes that it will store data in, so sample, variant, etc. And we can see here that so the genotypes are stored in 2 bits, which is a very small amount of data. So it's highly compressed, and it stores 2 alleles by, in this example, 2,504 samples by this many variants in this particular file. And just a note about the performance of GDS compared to other data formats. So this is a particular data set that I've worked with in the past from the TopMed project, which has about 18,000 samples and 219 million variants. And the file size for a BCF file is over 160 gigabytes. For a seq array file, you can get it down to less than 30 gigabytes. And then the read time to read data back in from the GTS file is also much faster. So if you were to read an entire BCF file, it would take you 182 minutes to read the 1,000 genomes phase 3 genotype data from a BCF file. It's only 16 minutes from a BCF, the binary call format file, but we can get that down to about 5 minutes with seq array. And these are some R functions that we're going to be using in this workshop to interact with data in a GDS file. So you can use this VCF to GDS function to convert a VCF file to GDS. There's also a conversion function for Plink bed files. So if you have data that's in Plink format, you can convert those as well. You can open and close the files, retrieve data, compute allele frequencies, and then set filters. And filters are important because they allow you to define a subset of samples or variants and get data from just that subset of data. Uh, so you can say you only want data from a few samples and then retrieve that information from your GDS file very quickly. And then the seq-var tools package has a number of additional functions that allow us to interact with the data. So we can read genotypes as in a somewhat more user-friendly format than the three-dimensional array using these get genotype functions. We can extract the dosage of the reference or alternate alleles. We can quickly read in important variant information and also do allele frequency calculations. The different distinction between these and the seq array allele frequency calculation is that the seq-var tools ones take into account allele counts on the sex chromosomes. So if you're working with sex chromosome data, it'll make sure that the allele counts are accurate for those. All right, so that's the end of our summary for how to work with GDS files, and now we're going to go through some actual examples in R. So I'm going to go to the GDS format section. And I'm going to show you the R code that we're going to use to interact with GDS files. So the first thing you're going to want to do in working with GDS files is to install all the packages you're going to use. These are the packages that are needed for the entire workshop. So you could, if you're going to go through all of the workshop exercises, you can install these all at once. We're going to use the tidyverse packages to do some data manipulation. Um, and we're going to install the BioC Manager package that will allow us to install Bioconductor packages. These are some suggested packages that are needed for Genesis for the specific types of association tests that we're going to do later on. We're going to install the Genesis package, which will install its dependencies also. And then for when we get to the variant annotation section, we're going to need these additional R packages as well with some variant annotations. So the first thing that we're going to need to do is to load the seq array R package. And then we're going to load the data that we want to convert from VCF to GDS. So all of the data that's used in these examples is available in the GitHub repository. So you can download this example data and work through all of these exercises yourself. So we have a VCF file this is 1,000 genomes data. This is chromosome 1 that we're going to use as an example here. So we're going to download the VCF file for chromosome 1, and then we're going to call this seq VCF to GDS function on the VCF file. We're going to give it the name of the GDS file that we want to have as output. We're going to tell it that we want to import the genotype field, and we're going to tell it what storage option we want to use. This is the most efficient storage algorithm that seq array can use, and I believe it's also the default. And so this function will then convert that VCF file to GDS for us to use in the rest of this workshop. So once we've converted our GDS file, we can open it using the seq open function. And if you just type GDS after you do that, you can see all of the data that's been imported into GDS. So we see that there are about 11,000 samples in this GDS file and also about 11,000 variants. This is just a small file as an example, so we can run through these exercises relatively quickly. It stores chromosome position, alleles, the genotype data. It also stores the phase of the genotype data. In some cases, when you're doing association studies, you usually don't need to worry about the phase, but if you're doing things like local ancestry analysis, phase can be important for whether the genotypes are all on the same, for each allele are all on the same strand or not. And then it has some annotation about the data that's extracted from the other fields of the VCF file. So there's the ID for the variant, the quality indicator, whether the genotypes are filtered or not, and then it can also have some additional spaces to include info and format fields, which are not in this particular file. So once we've opened our GDS file, we can use this seq get data function to read in information from the GDS file. So we can read in the sample ID, and here we see the 1,000 genomes identifiers that are the samples. We can read in the variant ID, and the variant ID in a GDS file is always an integer. So if you have like an RSID for your SNP, that's usually stored in this ID column under annotation, not in the variant ID. So you can look at the chromosome position, and here we're getting the RSID, which is this annotation /ID, is how you get the RSID. And we can see that some of our variants have RSIDs, but some of them do not. We can use the seq allele function to look at a histogram of the allele frequency. And this is the reference allele frequency by default. So most of the allele frequencies are near 1. We can also get minor allele frequency by using this minor equals true argument. And then we get this histogram where most of the minor allele frequencies are pretty low. So if we want to work with just a subset of data, we use this function called seq set filter. And you can specify the variance and the samples you want to include when you set a filter. So we're going to say we want to look at the first 10 variants and the first 5 samples. So once you've run this seq set filter command, um, then any subsequent call to a function that you call from the GDS format package or any of the packages it depends on, like Genesis, will only refer to the variance that you set in your filter. So if you call the seq get data function to return sample ID, it'll just give you the first 5 samples. And the same for variant ID, it'll give you the first 10 variants. And now we can look at the genotype data by calling seq get data genotype, and it'll return a 3-dimensional array because this is how genotypes are stored in seq array. Uh, so the first 2 variants it will tell you the uh, first dimension of this is the allele, and then the second dimension of this is the sample. So it gives you the genotype for each of these variants. Um, Working with 3-dimensional arrays can be a little bit challenging, so you can use the seq_var_tools package to get somewhat more convenient representations of your genotypes if you just want to look at them. Um, so you can use the get genotype function to read in all of your the variants as the alleles, the account of the reference allele. Um, or, and you can also read in variants by get genotype alleles will read in variants by character. So you can actually get the individual alleles for each genotype. And then you can also get dosage of alleles. You can get the reference dosage and the alternate dosage. Uh, And seq_var_tools has some additional functions for extracting variant level information. So you can look at the reference allele, the alternate allele. This variant info function is handy because it'll return the variant ID, chromosome position, ref, and alt for all your variants. Um, and so if we wanted to go back from looking at just a few variants to looking at all the variants again and all the samples, you can reset the filter. And now it'll tell you when you set the filter how many variants and samples that we're now looking at. And uh, we can look at how many alleles there are for each variant. So seq array can represent multi-allelic variants. So seq num allele will tell you the number of alleles for each variant, and we have a handful of variants that have multiple alleles. So you can look at these variants by finding which variants have allele count greater than 2. And then you can use this alt_char function to look at the alternate alleles for those variants. And here you see there's multiple alternate alleles for each of those variants. Uh, and then you can extract a particular alternate allele from this alt_char function by using the n argument to the alt_char function. You can also use this is_snv function to find out if your variants are SNVs or not. So it'll tell you, and you can use this argument for biallelic equals true to whether you're restricting your variants to just biallelic SNVs or not. And the last thing that you can look at for another way to look at your variants is to use this granges function. So granges is a core bioconductor package that represents variant information as ranges. And you can use these functions on a GDS object and extract information from your GDS file as a granges object. And here you can see that most of our ranges are just a single position because most of our variants are SNVs, but we do have some variants that are a range of positions because they're insertions or deletions. And then when you're done with this GDS file, you always want to close it with the seek close function. And these are some exercises that you can do later on to further explore working with GDS files. All right. And now we're going to move into how do we actually use these GDS files to do association testing. So we're going to go over the important functions in the Genesis package for working with GDS files. Uh, so our essential our objects for working in Genesis are the annotated data frame object. And this is an object that's defined in the BioBase package, which is one of the core bioconductor packages. And this object is an R data frame with metadata describing each column. So when we're working in Genesis, all of our data frames that are describing our sample data are going to be in this annotated data frame class. And this class is very handy because it allows you to easily share information about what actually is stored in each column of your sample annotation. So you can put things like the units that your outcome variables are stored in, uh, and various other information which you can use to communicate with your colleagues about your sample annotation, and also just to communicate with future versions of yourself. If when you're creating the sample annotation, if you're describing all your data, then when you go back and use it later, you always know exactly what the data is that you're working with. Uh, then there's a seekvar GDS class object, and this is the object that interfaces with the GDS file in the seek array package. On top of the seekvar GDS class object is a class called seekvar data, and what this does is an object that links a GDS file to the sample annotation. So the seekvar data object comprises both an annotated data frame and a seekvar GDS class object. And when you create this object, it makes sure that the samples in your annotated data frame match up with the samples in your seek array object. And then the final object that we're going to use is an object called a seekvar iterator that's also defined in the seekvar tools package. And this is an object that'll return the next set of variants with each iteration. And this is how we do association testing in Genesis by iterating through groups of variants. Since our uh, variant data is usually much too large to store in memory at once, we need to iterate through chunks of that data to do association tests on blocks of variants. And so that's what the seekvar iterator object will do. So the iterator object extends the seekvar data object. So all the methods that are defined in seekvar tools and seek array can be used on an iterator object. And there's a few different ways to define an iterator. So there's a seekvar block iterator, which just allows you to iterate over blocks of variants. This is the simplest type of iterator and probably the one you'll use the most frequently. Um, there's a range iterator that allows you to iterate over ranges defined by the chromosome, the start position, and the end position. There's a window iterator, which is a special case of the range iterator, where it defines for you a sliding window, where you can just slide your window across the genome and give you the next window every time you call the iterator class. And finally, there's a list iterator, which allows you to define specific lists of variants that you want to use. And we're going to use these objects when we get to doing aggregate tests like burden tests and scat tests. That's when these the lists and the range objects, iterator objects, become important. Uh, So here's an example of what you might actually want to do with this iterator object. So you might start with your phenotype data and your covariates, put those in an annotated data frame and store them in a seekvar data object. Um, you might use a seek set filter command to select only rare variants. You might group your variants then by gene using a seekvar list iterator. And then every time you call this function called iterate filter, it will return the next gene. And then you can run a burden test or a scat test on all the variants in that gene. All right, so these are the key functions in Genesis that we're going to be using to run association tests. So the first function is called fit null model, and that function fits a null model with fixed effects, and fixed effects are things like age, sex, smoking status, things that you might want to use as covariates in your analysis. And then it can also fit random effects, and random effects are going to be in the form of like a kinship matrix or a genetic relationship matrix to describe how your samples are related to each other. Once we've created a null model using the fit null model function, then we can call one of these two functions either a soc test single to run a single variant test, or a soc test aggregate to run an aggregate test on rare variants like a burden test or a scat test. So this is a flow chart of what your association test might look like. So you start out with your covariance matrix describing your sample relatedness. If you know that your samples are not related to each other, then you don't need to have this. You can run a Genesis function without this. But frequently, when you're working with study data, you might find that some of the samples in your study are actually, some of the people are actually related to each other, in which case you need to include this covariance matrix. Uh, you have an annotated data frame that has your phenotypes, and then you have your GDS file that has your genotypes. And so the kinship matrix and the annotated data frame both go into this fit null model function. The annotated data frame and the GDS both go into the seek var data function to create the seek var data object. And then you're going to somehow select what variants you want to use, and use that to define your iterator object using your seek var data object and your variant selection. And then you'll take the null model and the seek var iterator object, and you'll send those to either the associate test single function or the associate test aggregate function. Uh, And this is just a note on some of the more complicated parts of association testing. So if you have samples that have that are in different groups, say you have a cohort study and you've recruited your samples from different populations. Um, or you're combining multiple studies together into a single association analysis. It's sometimes the case that the variance in the different groups is not the same for your phenotype. And if that's true, then there's this additional additional parameter called group var for the grouping variable that you can allow the null model to fit heterogeneous variances by group. And so we'll see this in the exercises um, where we'll use this group variable to group our samples by study and allow the variance to be different by study. And this can often, if you have samples with different variances, this can reduce the genomic inflation in your association test. Um, there's also a more complicated procedure that you can do to do an inverse normal transform of your residuals. I'm not going to go into this procedure in detail, but this is a somewhat more involved version of doing an association test where you fit the null mix model multiple times in order to use in order to try to use the residuals as your outcome ultimately. And this can, if you're getting genomic inflation in your association test, this can sometimes help resolve that. So now we're going to go and look at actual code for running association testing. Uh, So we're going to start out by fitting a null model. And so the reason that we're doing this separately rather than just calling a single function to do the association testing is that especially if you're using a matrix like a kinship matrix or a genetic relationship matrix, it can be very computationally intensive to fit the null model. And you only need to do that one time. You don't need to do it for every single variant that you're computing the association test for, or every block of variants. So it's much more computationally efficient to fit that null model once and then take the results of the null model and use that for all of your subsequent variant testing than it is to compute it for every single block of variants that you're testing. Uh, So we're going to start with fitting the null model. We're going to need an annotated data frame that has our phenotypes. And so we're going to put that together based on, we have some sample annotation that's describing the samples with IDs that are matched to the GDS file. And then we're also going to have our phenotype file that has the subject ID, which is an ID that refers to a particular person. Um, Usually, we advise when you're designing a study to use separate identifiers for the samples, which are the sequencing or the genotyping instances, and the people with phenotypes, and maintaining a mapping file that matches your sample to your subject. And this is because sometimes mix-ups happen in the lab, and you can find out that the person that you thought you genotyped is actually somebody else. And it's very easy to correct that if you maintain a separate list of sample identifiers and subject identifiers, and it's much more difficult to correct it if you've just directly mapped your sequencing or genotyping instances directly to the identifier for a person. In this example, for the 1,000 genomes data, we're going to see that the IDs for sample and subject are the same. But if you're doing this in practice, we recommend using different identifiers for those. So we're going to read our sample annotation data. And so we've saved this as an RDS object, which is an R object stored on disk. And we're going to use this function read RDS to load that R object. And we're going to need to load the biobase package to work with annotated data frames. And we're going to take a look at this R object. So in an annotated data frame, you use the function pdata to extract the phenotype data from the object. And so we're just going to look at that, and we can see that it has information about the sample ID, the subject ID, um, the population for each of these 1,000 genome samples, the sex, and then we've just made up the status variable so that we can do an association test on it. Uh, And then let's say that we want to do an association test with for height, and that we want to adjust for some other data as covariates. So we're going to load some phenotype data, and this has also been stored in a annotated data frame object. And so we can see that we have subject ID, sex, age, height, and then the study that the data came from. And again, we've just made up this information. This isn't real data. This is for 1,000 genomes, but we invented it for the purpose of this exercise. Uh, and we can look at the metadata in our object using this var metadata function. And this is that useful information I was talking about earlier, where it'll tell you something about each of the columns in your data. So for example, we know that the subject's height is in centimeters by looking at the metadata for this object. All right, now we're going to load the dplyr package, which is part of the tidyverse, in order to join two data frames together. Um, so we're going to use this left join function from the dplyr package. So we're going to take the phenotype data from the annotated data frame, the phenotype data from the phenotype data frame, and join them together. And we're going to join on the subject ID and the sex, because those should both be the same between the two different files. We're also going to join the metadata together by just binding the rows of the two annotation objects. But we need to only take columns 3 to 5 for the phenotype annotation because we don't want to repeat the annotation for subject ID and sex. So we just want the annotation for these 3 columns. And so we're going to save that into a new annotated data frame, and then we'll save that as an RDS object. So once we have our new annotated data frame, we can load the Genesis package. and call the fit null model function on this package. Uh, and we're going to pass it the annotation object. We're going to tell it that the outcome is height. And we're going to um, we're going to tell it which covariates we want to use. So we're going to say sex, age, and study are the covariates in our analysis. Um, we are going to use study as a grouping variable, like we talked about earlier. Um, and then we'll save this null model object as an RDS file. And if you want to do the more complicated procedure of taking an inverse normal transform and refitting the model, there's some documentation here about how to do it. In the interest of time, I'm not going to go through all of the details in that, but there's a pipeline that I wrote several years ago that uses Genesis to run association tests. And the documentation for that pipeline has information about how to do the inverse normal transform. So now once we've created this null model, now we can actually move to running an association test on our genotypes. So we're going to need to load the seqvar tools package. And then we're going to need to open our GDS file. So we're going to use this chromosome 1 file as an example. We're going to open our GDS file. And then we're going to call the seqvar data function, which creates a seqvar data object from our GDS file and our sample annotation. And what it does when it creates this object is it checks to make sure that the sample IDs in this object match the sample IDs in this object. And now that we've created our seqvar data object, then we need to define how we're going to iterate through our variants. And for our single variant association test, then we're going to use the seqvar block iterator. So to create one of those, you just pass it the seqvar data object. And once we've created that iterator, then we call assoc test single, and we just pass it the iterator object and the null model object. And then it'll tell you how many samples it's running on. It'll tell you something about the that it's using 8 cores to run in parallel. Uh, and then it will return to you the association results for each variant. And so this is what they look like. You get the chromosome position and variant ID. If there are multiple alternate alleles for a variant, this allele index could be greater than 1 because it'll test each alternate allele for each variant separately. It'll tell you the number of observations it used. This is generally equal to the number of samples, but if you have missing data for any of your samples, then it might end up being less than the number of samples because it can't run the test on missing data. It'll tell you the frequency of the alternate allele, the minor allele count, and then it gives you the results for the test itself. There's a few different options you can use for testing, but the default is to run a score test. So it'll give you the result of that score test, the standard error, the test statistic, and the p-value. And then it also does an estimate of the beta and the standard error of the beta. And it will also return the percent variance explained for each variant. And so then after you've done this, you might want to make a QQ plot to examine the results. So this is just an example of creating a QQ plot function using the ggplot2 package. And so we can make a QQ plot of our p-value. And we see that there's actually quite a lot of inflation here. So you want most of your points to be on this red y equals x line with only sort of the tail of the distribution veering off this line to show that you have done a good job of controlling for your inflation in your test. And it looks like we have it here. This is probably because we did not account for any relatedness between our samples. So we did not include a kinship or relatedness matrix when we ran our tests. So when we get to the next section on population structure and relatedness, we'll actually see that we can start doing better in our association testing, controlling for inflation. And there are some exercises that you can go through. So we just did an example of quantitative phenotype, but you can also use logistic regression for a binary phenotype. Um, so I encourage you to try these exercises out on your own in the next week before the next session. And then the other type of test that we can do besides a single variant test is a burden test or a scat test. And the simplest way to collect variance for running a burden test or a scat test is to do a sliding window across the genome. Uh, so uh, we want to reset our filter to make sure that we're including all of our data. So when you create an iterator object, it will set a filter on the data to loop through different blocks of variance. So if you run one association test already in your R session, and you want to start a new analysis, you always want to reset your filter. And then we're going to create a sliding window object with the seek_var window iterator. And there's a couple of arguments you can give it of how big you want your window to be and how big the shift should be. There are some default values for these in the function. And once you've created your sliding window iterator object, you can call associate_test aggregate on your iterator, the null model that we created earlier. We're going to run a burden test. And we're going to set a maximum allele frequency. So usually when you're running a burden test or a scat test, you want to restrict to rare variance because the idea behind these tests is that you might have a number of rare variants that are all contributing a little bit to your association. And when you consider them in aggregate, you might be able to detect an association that you wouldn't find for any individual variant by itself. And the idea behind the sliding window is that if you have multiple variants contributing to your effect, they might end up being close to each other in the genome, say for like from the same gene. Usually you would set this a maximum allele frequency to be less than 0.1, but since we are using example data that doesn't have a lot of variance in it, we have to set it to 0.1 to get enough variance in our test. So when we call associate_test aggregate, um, it gives you a list of 2 data frames in this case. It gives you the results of your association test, and it also gives you variant info. And so the results of the association test are similar to what we just saw for the single variant test, but instead of individual variants, it'll give you the start and end position of each of the windows that it tested. Um, and then it'll tell you the number of variants that were included in the test. And since this data is very sparse, it's just one variant per window. It'll give you the number of alternate alleles that were observed, and then the number of samples that have alternate alleles in each of those windows. And then you can also use this association variant info object to look at which variants were actually included in each of your sliding windows. And again, you can make a QQ plot of the association results. And again, we see that the results from this test are inflated because we haven't properly accounted for the relatedness between our samples. You can do another type of test that's called a SCAT test for sequence kernel association test. Uh, and So for this test, we're going to reset our filter. We're going to define a new seek_var window iterator. And then we're going to call the test equals scat. And then we're going to, for this weight beta object, we're going to use what's called the woo weights. And this is just a way that we're weighting variance in our sliding windows. So usually when you run a burden test, you want to set your weights to be 1, 1. And when you run a scat test, you want to set your weights to be 125. And there's more information about what exactly this weighting function is doing in the documentation for the SOCH test aggregate function. And this is the results for the scat test. So it has the same information about each of the windows, but the test statistic it gives you is slightly different. It's called Q. It gives you a p-value, an error, and then it also tells you what what method it used to calculate the p-values for scat. We can again look at the variant info for all of our windows, and then plot the p-value results. And for an exercise, you can try to repeat this using logistic regression instead of uh, the quantitative trait. And I'm going to pause here before I go on to the next section to see if anybody has questions.
Thanks, Stephanie. There are a couple of questions on the Framapad. Would you like me to read them out?
Sure. I can also try
Yeah.
to pull that up.
All right. So the first question, how many would you like to take at this point before you continue? Because we also have 10
Let's see.
minutes left.
Yeah. Well, I guess I'll just see if I can answer these one at a
Zero
time.
genome.
So it looks like someone is asking, does Genesis work for a Dart-seq dataset? Um, I'm not familiar with Dart-seq data, so I do not actually know the answer to that question. Um, if your data is in a VCF file or a Plink file, you should be able to convert it to seq array. But since I'm not familiar with that type of data, I can't answer beyond that. I'm sorry. Uh, and someone's asking, what's the minimum number of samples to do an association analysis? Um, That's more of I think, a theoretical question for when you actually have enough power to detect an association. And I think you can use any number of samples in Genesis and get a result, but you might not actually be able to detect an association if your sample size is too small, because you might not have enough statistical power to detect that association. Can you use Genesis for non-model organisms? Yes, you can. Uh, and can you provide your own annotation to Genesis? Yes, and we're going to cover some variant annotation topics next week. Does GDS format and the tools work for polyploid data? Yes, it does. So if when the genotypes are encoded in seq array, it has a number of alleles that's usually two for human organisms. But if you have multiple alleles for a non-human organism, and that's encoded in your VCF file, you should be able to convert that to seq array as well and use those GDS files in Genesis. Someone's asking about what to do next for a first-time researcher working on GWAS. Um, so I feel like this question might be a little bit broader than I can answer in the next few minutes. Uh, a workflow so as to target high impact journals. I can refer you to, there is a pipeline that I wrote to do association testing, which is, I can point you to the GitHub repository for that. Uh, oops. So this is a pipeline that I'm going to put it in the chat. Um, does association testing on sort of this was designed for um, genotype data with a very large number of samples and variants. Um, and it has a README that steps through running all of the different steps in the association test and how to set up the pipeline. So this might be a good example of a workflow beyond what the simple example that I'm presenting here. This workflow is sort of how you would proceed if you had a larger amount of data and it takes you through all the steps of doing the association test. Someone's asking if I could assist in doing analyses. So if you have questions about using the code, I'm happy to try to answer those. There's a Bioconductor support website, and if you tag questions with Genesis on the Bioconductor support website, I will see them. You can also open issues on the Genesis um, uh, GitHub repository. So if you have questions on specifically how to use the package, then I can help. I can't help with sort of more general, how do you design your experiment questions? That's sort of beyond what I have the capacity to help with, but I can help with any issues that you have running the code. Uh, can you use HapMap formatted SNP data directly in the Genesis package? Yes, you can. Someone's asking about not incorporating PCA due to the absence of population structure. Um, the next section is on running PCA and including population structure. So what we saw in our QQ plots is that the we had some inflation off of the y equals x line, and that's actually because we're not accounting for population structure in this analysis, and we should be. So the next section is going to be how to include population structure in our analysis. Um, what's the maximum number of PCs that can be incorporated in the analysis? There is no maximum number. I've done an analysis recently where we incorporated like 35 PCs because we had data that had a lot of population structure. And so we wanted to include a large number of PCs. In the next section, we're going to look at how to assess which PCs you should be including, because you generally want to assess which PCs are informative for your particular analysis, and then use that to decide how many to include. Let's see, it looks like there might be some questions in the chat as well. Someone's asking how you get genotypes. Um, I think that's a question that's beyond the scope of this workshop, but if you want to just start testing out the packages, you can use Thousand Genomes data, which is publicly available. And if you're wanting to actually do a new analysis, then you would need to connect with a genotyping center or a sequencing center to actually get genotypes or sequences from someone from a cohort of participants. All right, I think that's all the questions.
Yes, that's all the questions for now.
Okay. And we're almost at the end of the hour. So I'm not going to start on the next section just yet, but I'm going to give you a preview of what we're going to cover next week, which is we're going to start talking about population structure and relatedness and how you include that information in your association test, which is running a mixed model. Um, we're going to talk a little bit about variant annotation and how you can include variant annotations in your tests, and then run aggregate tests based on that variant annotation. I also wanted to highlight some background information that might be useful. So I haven't talked at all about the theory of GWAS, since this is just a practical workshop for how to run GWAS in R. Um, but if you want more theory of GWAS, there are slides here prepared by one of my colleagues on the theory behind genome-wide association studies. There are also some slides that I presented in a previous workshop that I did a few years ago, along with a video of me talking through those slides that you can watch that provides some background information on variant annotation. I'm just going to jump right into the practical aspects of using variant annotation next time. So if you want some background information, you can watch this video in advance of the next class. Um, and then there's also a sort of much longer 2.5 day long um, uh, workshop that I presented back in 2020 during the pandemic. And since it was pandemic time, all the videos were recorded and posted online. So some of the material from that is what I talked through today, but there's some videos of me presenting that. And then there's a lot of theory behind GWAS, population structure, and there's more details about various aspects of GWAS. So you can look through the website for that class and watch those videos as well if you're interested. And I guess we're at the top of the hour, so I will wrap up for today. Thank you for attending, and I'll see you all next week for part 2.
Thank you so much, Stephanie, for that presentation, and everyone else for attending and staying this late to attend the session. We will have another session next week at the same time. And I reckon if you've registered for this one, you should get an automatic link to the next session. But in any case, if you do not get it, I will be sharing a feedback form tomorrow, and I will include a link to register for that one as well, in case you don't get it. The link to the materials that Stephanie has been sharing is on the Framer pad on line 59 and line 60, if you'd like to go through that in preparation for the next session. Yes, thank you so much, everyone, for attending. I don't know if Maria would like to say something because she's our community manager at Bioconductor.
I'll just be very quick and just say thanks very much, Stephanie, for an excellent session, and thanks everyone who joined today. Yeah, we really appreciate your time and engagement. We look forward to seeing everyone next week for the second session. So have a great day, everyone. Bye.
Thank you. Goodbye. See you next week.